Welcome, colleagues, fellow researchers, scientists, and students alike. Thank you for joining. This is Microbial Mutation and Drug Resistance, a scientific research series developed and presented by Bryan Geniuson, research scientist, founder, and research systems architect at Natural Scientifics. This opening installment establishes the conceptual and scientific foundation for the entire series. Drug resistance is often described as a failure of medicine. Biologically, resistance is not a failure. It is an outcome. It is the predictable result of applying linear pharmacological strategies to adaptive, self-modifying biological systems. Understanding drug resistance requires moving beyond the question of why microbes outsmart drugs, and instead examining how molecular biology, pharmacology, and biological systems respond to survival pressure. Microbial drug resistance does not emerge from a single mutation, a single gene, or a single exposure. It emerges from systems-level interactions between genetic variation, molecular signaling pathways, cellular stress responses, population dynamics, ecological structure, and selective pressure imposed by therapeutic agents. This series approaches resistance not as a molecular accident, but as a systems behavior rooted in molecular biology. At the molecular level, antimicrobial agents exert selective pressure by targeting specific biological structures and processes, including cell wall synthesis, nucleic acid replication, transcriptional machinery, ribosomal function, and metabolic enzymes. These pharmacologic targets define both vulnerability and evolutionary constraint. All biological replication systems operate with imperfection. Even under stable conditions, DNA polymerases introduce rare copying errors. In large microbial populations, probability guarantees that molecular variation will exist. Most mutations are neutral, many are harmful. A small fraction alter protein structure, enzyme kinetics, target binding affinity, regulatory control, or membrane dynamics in ways that become meaningful when pharmacologic pressure is applied. Mutation does not create resistance, it creates molecular possibility. When survival is threatened, fidelity becomes secondary. Under antimicrobial exposure, immune pressure, oxidative stress, or environmental insult, microbes activate conserved stress response programs that alter transcriptional regulation, DNA repair fidelity, protein synthesis pathways, and metabolic flux. Replication continues even when accuracy must be sacrificed. From a systems biology perspective, mutation rate itself becomes a regulated molecular variable rather than a fixed property. Selection does not create resistance. Selection amplifies molecular variants that already exist. Any variant that reduces drug binding, alters target conformation, increases efflux capacity, modifies permeability, or reroutes metabolic pathways gains a disproportionate survival advantage under pharmacologic exposure. Through rapid replication and population expansion, rare molecular variants become dominant lineages. Resistance, therefore, emerges not through intention, but through pharmacodynamic selection mathematics. Resistance is not confined to vertical inheritance. Through horizontal gene transfer, including conjugation, transformation, and transduction, resistance determinants such as enzymes, transporters, regulatory elements, and protective proteins propagate laterally across microbial communities and species. Once resistance enters a system, it becomes a networked molecular property. Higher-order biological structures further stabilize resistance. Biofilms, persister states, and microbial ecosystems alter drug diffusion, local concentration gradients, metabolic state, immune visibility, and target accessibility. These structures create pharmacologic refuges that allow resistant and tolerant populations to persist even in the absence of continued drug exposure. However, important scientific limitations and challenges must be acknowledged. Current resistance models often rely on reductionist assumptions that isolate single genes, single targets, or single organisms, despite resistance emerging across interacting molecular, cellular, population, and ecological layers. Experimental systems frequently fail to replicate real-world complexity, including fluctuating drug concentrations, host-immune interactions, microbiome context, and spatial heterogeneity. Pharmacokinetic and pharmacodynamic models remain incomplete predictors of evolutionary outcomes, particularly under chronic or sublethal exposure. Genomic surveillance can identify resistance markers, but often lags behind phenotypic adaptation and emergent tolerance states. Predicting evolutionary trajectories remains constrained by stochasticity, nonlinear feedback loops, and incomplete mechanistic resolution at scale. These limitations do not invalidate current science. They define the frontier. Most antimicrobial strategies are designed to inhibit a single molecular target or pathway. Resistance, however, does not exist at a single molecular scale. It exists across interacting layers, molecular targets, cellular physiology, population dynamics, ecological systems, and therapeutic environments. When linear pharmacologic interventions confront adaptive molecular networks, resistance is not an anomaly. It is the expected result. This series is grounded in a systems biology principle. Biological systems do not fail under pressure. They reorganize at the molecular, cellular, and population levels. Drug resistance is not opposition to therapy. It is the biological system's adaptive molecular response to pharmacologic stress. The MMDR series exists to map the full architecture of microbial drug resistance, from molecular mutation and pharmacologic targeting to ecological propagation, using a systems biology framework aligned with evolutionary and biochemical reality. Drug resistance is not mysterious, it is logical, and once understood as a molecular, pharmacologic, and systems-level phenomenon, it becomes addressable as one. A fully referenced scientific literature packet supporting this episode and the complete MMDR series, including peer-reviewed studies, mechanistic analyses, molecular biology references, pharmacologic models, and curated source annotations, is available as a downloadable companion resource for purchase. Access and purchasing information is provided in the video description. This content is provided for educational and scientific discussion purposes only. It does not constitute medical advice, diagnosis, or treatment.